Robert.
Thank you very much. You've stolen my first slide, actually. That's what I have here. As Andrew mentioned, we've had some brilliant talks showing the role of diet in child nutritional status. And I guess I'm here today to try and argue that there's another facet of child development which may play a really significant role in early child growth, namely the microbiota or the microbiome. And so the basis of this talk is, as you mentioned, that what I'm going to try and persuade you is that we need to shift away from the pathogenic model of child undernutrition. I'm not denying, of course, there is a huge role for pathogens and infection in impairing child growth, but a microorganism isn't necessarily always a pathogen. And there's trillions of other microorganisms inside the human body which play many more important roles for benefits, which may go against what pathogens might be doing. And that's what I hope to kind of persuade you and describe to you today. So first, I'm just going to run through what we already know about the microbial role in child malnutrition and the role for pathogens, and Andrew has touched on some of that already. I'll give a little introduction to the gut microbiome for those that aren't as familiar with it, and then I want to bring you through the chronology, I suppose, of the acquisition and the succession of the gut microbiome in the child over the first 1,000 days, and if that becomes disturbed, how that may affect the pathways relating to growth. And finally, I want to then discuss some of the evidence that's already there for interventions that may target the microbiome and where we could move in the future to propose some of these interventions which may go hand in hand with nutrition and WASH and some of these other environmental interventions that we've spoken about. So first, what do we know about the microbial influence in child malnutrition? I don't need to go through these stats again. We've seen them in a few lectures already. We know that nutrition interventions aren't good enough to completely restore child growth. We know that the very best nutritional interventions will only restore stunting by 20% and wasting by 60%. So there's a huge number of other environmental factors which contribute to faltered child growth. One of which could be the acquisition of microbes in very early life. And we know that the carriage of enteropathogens is almost ubiquitous among children from low resource settings. So here's a couple of children, one who has edematous malnutrition, one who has more classical wasting. We almost know for sure that these kids will be carrying at least one enteropathogen, even if they present with a healthy nutritional status from a very low resource setting. Nearly all kids are going to be carrying at least one enteropathogen. And a majority of the time this is subclinical. They're not going to present with a really intense infection, but if you screen for it, they will be carrying something. And even if these pathogen carriage is subclinical, these pathogens contribute to faltered growth. And we know this from huge cohort studies such as MALI-D, where the higher carriage or the higher number of pathogens that a child is carrying is inversely related to linear growth. And we can see that it contributes quite a lot, maybe up to half a Z-score of reduced linear growth. And the pathogens that are contributing most to this faltered growth are usually those involved in disruption of the mucosa, such as Giardia and a few others. And we also know that treating kids with antibiotics does have growth benefits. In this meta-analysis by some colleagues of ours, they found that in a meta-analysis of randomized controlled trials, treating children with antibiotics led to an average increase in weight of 23.8 grams per month and a small but significant increase in linear growth as well of 0.04 centimeters per month. So clearly antibiotics are targeting some sort of infection, even if it's subclinical, in the body, which may be somewhat restoring some of this growth that we see in undernourished children. So that's what we know already about the pathogenic model of child malnutrition. We know that infection and pathogen carriage are extremely common in children who are stunted or with more severe wasting. We know that antibiotics improve growth, even in children who don't have severe malnutrition. And we know that pathogens don't fully explain growth faltering, just as diet doesn't fully explain it. If we treat these pathogens, then it does lead to small improvements in growth, but it doesn't fully restore that full growth potential. And what we know from westernized chronic intestinal diseases is there's also this enteropathy. Excuse me. There's an environmental enteropathy in child undernutrition, which is similar, although not exactly the same as the enteropathy we'd see in some chronic Western intestinal diseases, where there's this blunted villi, local and chronic inflammation, epithelial shedding, and general impairment of that intestinal barrier. And in Western intestinal disease states, we know now from lots of microbiome sequencing studies that there is a microbial influence on this enteropathy. So we'd hypothesize that the microbiome in child undernutrition may contribute to some of this environmental enteropathy as well. So I want to introduce what this collection of microorganisms is in the gut, which aren't necessarily the pathogens. Many of these are commensal microorganisms, which are extremely important for training the immune system in early life. But some of them are what can be termed pathobionts, where in a healthy state, they play no harm in the body. And then if something occurs environmentally, this can trigger it to become a pathogen, much like the H. pylori story that we've spoken about. So when we talk about the microbiota and the microbiome, these terms are often used interchangeably these days, but strictly speaking, the microbiota is the compositional collection of microorganisms in a given environment, in this case, the human body. And one of the well-known facts of this field is that we are outnumbered by microorganisms in the human body. And it's incredible to think that the number of cells in the human body are more microbial than they are mammalian. And if we look at the genomic potential, or the number of genes in the body, we're outnumbered almost 100 to 1 by microbial genes. So you can see there's huge functional potential there for the microbiome to manipulate host physiology, especially in vulnerable states, such as in child undernutrition. And if we view or image the microbiome, we can see that different species like to live in different regions of the intestinal tract. Some of these species prefer to live right down in the mucosal layer, taxa such as Akkermansia, mucinifera, which feed off the mucus, whereas many others prefer to reside a bit higher up in the lumen, where there's more abundance of nutrients being passed down. So within the human body, within the different niches of the human body, there's different composition of microbes, but even within the intestine itself, in those little localized regions, there can be quite a huge variation in the types of microbes that live there. And when we focus on the gut microbiome, for years, and we continue to do so, we rely on stool as a proxy for what is living inside there. But some recent data from Erin Ellenav's group has shown that this mightn't be quite a suitable proxy for what's going on in the gut. The different regions of the intestines contain drastically different makeup or composition of at least bacterial microbes. And as I've mentioned in the previous slide, there are drastic differences even between the microbes residing close to the mucosal layer as opposed to those living in the lumen. So although stool is a lovely non-invasive way of studying the gut microbiome, it's definitely not representative of the entire intestinal tract. And when we talk about phenomena like environmental enteropathy, which predominantly occurs in the upper intestinal tract, stool may not represent exactly what is going on there from a microbiome perspective. So the functions of the gut microbiome in the child are way too vast to put onto one slide, but to summarize in a way, an infant is exposed to this huge coating of microbes as soon as they're born. And this plays an extremely important role in the training and the maturation of the immune system in early life, and teaching the immune system to learn what is a pathogen and what is a commensal microorganism. But this vast collection of microbes also produce a huge number of metabolites as well, particularly short-chain fatty acids, which are byproducts of carbohydrate digestion, and things like secondary bile acids as well, which play an extremely important role in nutrient digestion. The gut microbiome can play a huge role in nutrient digestion as well, and this is extremely important when we consider infants who may not be able to absorb and acquire food as well as a healthy child. And so what I want to persuade you today is the microbiome may play a big role in this nutrient absorption. And finally, as I've mentioned, there's an influence of the gut microbiome in maintaining the integrity of the epithelial barrier and preventing some of this translocation of LPS and gram-positives that we've spoken about before into the bloodstream, which may contribute to some of this chronic inflammation, which contributes to faltered growth. Oh, the slide hasn't appeared very well, but what it's supposed to show is that when we consider the microbiome, it's hugely context dependent. The gut microbiome is geographical. A child who's living in India or in Bangladesh has quite a different microbiome to that living in Malawi, for example. But we know from studying a westernized populations versus those, for example, in a rural population in Venezuela who've uncontacted hunter-gatherer tribes, that their microbiomes drastically differ, as you might expect. And it's hypothesized that the chronic exposure to antibiotics, Western diets, and lifestyles are what contributing to this separation or this loss in commensal organisms. But even looking at differences between, for example, as I said, India or Malawi or some of these other populations, there can be quite drastically differences in the composition of the microbiome. So this really needs to be considered when we're studying its effects in child growth. So as I mentioned, I'd like to take you through a walkthrough of the first 1,000 days and how the gut microbiome is acquired in a child from birth and possibly even before birth, and how that may contribute to faltered growth if this acquisition and succession of a microbial organ is disturbed. And there are a huge number of factors which can influence this composition during the first 1,000 days. There's some interesting data to show that the maternal microbiome may even affect prenatal growth, namely preterm birth and intrauterine growth restriction. So we know that bacterial vaginosis and periodontitis are associated with preterm birth and possibly even reduced birth weight. So there may be a microbial influence from the mother on the infant before the child is even born, which may affect their growth trajectories in early life. And then after birth, the influence of antibiotics, of genetics, of breastfeeding status, and even birth delivery mode play a huge role in the types of bacteria and viruses and other microbes that reside and colonize the infant intestinal tract. So what happens during pregnancy in the maternal microbiome? I'd originally named the talk the gut microbiome in child malnutrition, but decided to change it to the human microbiome because there are other ecological niches within the human body, particularly in the pregnant mother, which may affect growth of the child. And we know this we hypothesize this from some studies in Western settings. So we know that preterm birth is more prevalent in mothers with IBD. And IBD has a huge influence from the gut microbiome. We know that antibiotics improve birth weight and reduce preterm birth in low and middle income settings. So therefore, there may be some sort of microbial influence on growth, and by removing those pathogens, this can maybe improve the growth of the child prenatally. We know that gut microbial translocation increases during pregnancy. So if a woman is susceptible or is carrying enteropathogens, this may trigger a chronic inflammatory disease state, which may reduce the growth of the child or its full potential. And we know that maternal periodontal infection, as I've mentioned, is associated with preterm birth. So when we talk about translocation across into the bloodstream, this doesn't only occur in the gut. There's interesting evidence to suggest this may happen in the oral cavity as well. And this has been shown for some fascinating studies of the placental microbiome. And sequencing the microbes that are in the placenta, it's been found that this microbiome is most similar to the maternal oral cavity, which lends to the hypothesis that the maternal oral cavity may act as a storehouse of microbes that may seed or at least indirectly interact with the fetus, even prenatally. So the vaginal microbiome seems to anticipate pregnancy, seems to anticipate birth. Where during pregnancy, if you sequence the microbiome during pregnancy at a number of different time points, it remains fairly stable in its diversity. And then until about 40 or 50 days before pregnancy, suddenly there's a rapid increase in the diversity of microbes that are in there. So it seems that there's a kind of evolutionary adaptation of the pregnant female body to prepare the microbes that reside in the birth canal to interact with the child when it's born. And it was commonly thought for the last 10 years or so that the vaginal microbiome is predominantly made up of one of these 4 lactobacilli species. If you sequence the microbiome from a woman, the vaginal microbiome from the US or the UK or a Western country, 99% of that will be one of these 4 lactobacillus species here. But there's a small group of women who have a more diverse microbiome, and these have been classified as community state types. And this community state type number 4 here, which isn't defined by one of these lactobacillus strains, is hugely associated with preterm birth in the US at least. So there isn't bacterial vaginosis occurring here. There's merely a shift in the composition of the microbiome during pregnancy, which may be contributing to preterm birth and, of course, to fetal growth as well. And so it was thought that this occurs all over the world, and the vaginal microbiome everywhere is made up predominantly of lactobacilli. But a study by some colleagues of ours in London who conducted a huge trial of the vaginal and the placental microbiome in Malawi found that this wasn't the case whatsoever. In fact, only a very, very small number of mothers had a lactobacilli-dominant vaginal microbiome in Malawi. And instead, these community state types were defined by other species. So many of them were made up of Gardnerella vaginalis or some other predominant species in the microbiome. But similarly, in the Western disease settings, this more diverse vaginal microbiome was associated with preterm birth and with nutritional status of the infant. And we can see that here. So it's not only preterm birth. If a woman had a vaginal microbiome which was defined as this community state type number 4, which was very diverse, this was associated with a newborn length for age Z score, almost 0.4 standard deviations less than the other children, the controls, and also about a week less of duration of pregnancy. So again, this isn't driven by a pathogen. It's driven by a shift in the composition of the microbiome in the vagina, which may affect the growth of the child prenatally. So we move on to after birth then, and how does the infant microbiome develop in the first 6 months of life, and how does that contribute to growth? Well, the infant microbiome is predominantly driven by breastfeeding. Breastfeeding is the biggest contributor to infant microbiome variation in the first few months of life. Mode of delivery plays a huge role, so children born by C-section have quite a different microbiome to that of those born by standard vaginal delivery, but this tends to be corrected by breastfeeding after a couple of months. And a study a couple of years ago suggested that the milk microbiome, or the microbiome of the mother's milk, was quite rich, and that it might contribute up to 30% almost of the infant microbiome. So not only is the child being seeded by the birth canal and the vaginal microbiome, it's not only been seeded by the skin and the outside environment, but there also appears to be microbes within the breast milk which are contributing more to the infant gut than the skin. And so this is what happens in the first 6 months of life. The infant microbiome is predominantly made up of a couple of different species, usually bifidobacteria, which contribute sometimes up to 60-70% of the infant microbiome in the first 6 months of life if the child is exclusively breastfed, and also Bacteroides. And these two species are the ones that are highly, highly efficient at digesting and breaking down human milk oligosaccharides, this complex array of sugars which are present in breast milk. And so the infant microbiome tends to remain not very diverse at all in the first 6 months of life if the child is exclusively breastfed. It's predominantly made up of these anaerobic species which degrade the HMOs until the onset of weaning, which I'll speak about in a minute. And so the interaction between these milk oligosaccharides and the microbiome may play a really important role in child growth, as we've seen from studies from Jeff Gordon's group. So if we take if we profile the HMOs in a mother's milk, we'll see that those that have a stunted child produce less HMOs than those that have a healthy child, as we can see here on the left. And then if you genotype the mother for her secretor status, looking at the FUT2 gene, this variation in HMO production is widened between those that produce healthy and stunted children. So even there's a maternal influence in I suppose, the nutrition that a child is getting in early life. And because these HMOs require the microbiome to be digested, there's a really important interaction going on here. If you transplant the microbiome from a stunted child or a non-stunted child into germ-free mice, you can transfer this phenotype as well, which I'll speak a little bit about. But this stunted phenotype requires the interaction between the milk oligosaccharides and the microbiome, whereby if you supplement a mouse with structurally similar bovine milk oligosaccharides, and the mouse doesn't have a microbiome, it won't recapitulate growth. These HMOs require degradation by the microbiome in order to elicit this normal, healthy growth, at least in mice and in pigs from this study. So that's the milk-orientated microbiome, what happens in the first 6 months of life. But from 6 months onwards, weaning begins, and the child is exposed to a huge number of nutrients which feed the microbiome. It's been seeded in the first 6 months of life from breast milk and from exposure to the mother. And from now on, once the child starts eating, it begins to feed some different species within the gut. So we come back to this trajectories of the microbiome. And within 6 months and 2 years, there's these huge fluctuations in some of these other species here. The species like bifidobacteria tend to decline because they're not being fed by the HMOs anymore in the milk. And some of these other species which degrade some of the complex polysaccharides in plant foods suddenly begin to bloom. So species within the Lactobacillus family and Ruminococconaceae family. And these degrade all the complex polysaccharides we'd find in plants and produce some of these metabolites such as short-chain fatty acids. So there's big fluctuations in the microbiome from the point of weaning onwards up to about 2 or 3 years of life. And a study only out last week from the Pasteur Institute characterized what they call this weaning response. in an animal study, where they showed that the microbiome, as I've said, tends to be quite low in diversity in the first 6 months of life because the only source of nutrients is usually from breast milk. But suddenly upon weaning, there's this rapid expansion in microbiome diversity, and this happens concurrently with a huge rapid expansion in the immune responses and the training of T cells and Tregs, which they've characterized a bit more deeply here. But if you dampen this microbiome weaning response with antibiotics, if you prevent this big bloom, there are huge deficits in this immune response, and at least in animals, the child becomes more prone to infection and more likely to die or show a bad immune response to infection. So this expansion in microbes upon weaning is really, really important for the training of the immune system in the child, which, as we know, plays a very important role in growth. So that's what we can see here. After 2 or 3 years of life, after this 1,000-day period, the microbiome tends to be set in a child. The microbiome of a 2 or 3-year-old child is very, very similar to that of an adult. So when we talk about the first 1,000 days and the trajectories of immune development, of cognitive development, and of growth during this period, I'd argue that we also need to consider the microbiome development during this period as well, because there are huge fluctuations during this period which may set these trajectories of child development, which tend to plateau then after 2 or 3 years, and it tends to be set not permanently for the rest of life, but it tends to be a fairly steady state. So what happens then what do we see in child malnutrition, and what do their microbes actually look like? Well, we know from animal studies at least that the microbiome is critical for normal growth. If you take a germ-free mouse, it doesn't grow as well. This is a mouse that's been born by C-section, brought up in a bubble, is essentially sterile. It doesn't grow as well in its length and its weight as a conventionally colonized mouse. But if you monocolonize these germ-free mice with a single Lactobacillus plantarum species, you're able to recapitulate that growth. And I won't go into the details of this, but this is a paper from Science a couple of years ago that showed that this lactobacilli, which we've seen before in the maternal vaginal microbiome, interacts with the processes leading to growth hormone production and IGF-1 production. So a single strain itself can recapitulate growth deficits associated with being germ-free. But we know that just like nutrition doesn't act alone in growth faltering, the microbiome doesn't act alone as well, and it needs that interaction from between the diet and between the microbes, the commensal microbes that live in the gut. And this was another animal model of EED and growth faltering in mice, where we see this normal intestinal function and structure in a healthy mouse. If you feed that mouse a malnourished diet, there's a small change in the intestinal structure and function. If you instead give the mouse this cocktail of microbes, which aren't necessarily pathogenic, they're what I'd maybe term as pathobionts, there's a similar, not much of a change in the structure and function, but if you feed both together, the undernourished diet and this cocktail of microbes, there's this classic pathology of EED, this epithelial shedding, impairment of the mucus, and general impairment of the epithelial barrier. So there's this interaction and combined effect of an impaired diet and an impaired or dysbiotic microbiome, which needs to occur in order to elicit some of these pathologies associated with undernutrition. And this is the kind of seminal study which I was speaking about earlier, which took a cohort of twins from Malawi a good few years ago now, which were discordant for severe acute malnutrition, where one of the twins was healthy and one had SAM. And they sequenced the microbiome from all of these children and transplanted the stool samples into these germ-free mice. And if you transplanted the healthy microbiome into one group of mice and the malnourished group of microbiome into another group of mice and fed them both the same undernourished diet, this phenotype of malnutrition was transferred, which suggests there is some sort of causal role for a disturbed or malnourished microbiome in faltering growth. And this appears to be at least temporarily recovered with RUTF feeding, although not fully so. So the same group then tried to characterize this in a child cohort in neighboring Bangladesh and try and find out what does the normal microbiome look like in a healthy growing child during this first 1,000-day period. And in order to do this, they took a relatively small group of infants who were growing normally and healthily up to a normal length for age Z score, and I think a weight for height Z score as well, and took a stool sample every month and sequenced it to try and identify the species within the gut which were contributing most strongly to normal growth. And using some machine learning methods, they managed to identify a collection of 24 microbes which were contributing most strongly to normal child growth in this first 2-year period. And you'll see up the top here some of these species which are important during breastfeeding. For example, Bifidobacterium longum was really contributing a lot to healthy child growth. And so from this, the authors managed to create what they dubbed a Microbiota-for-age Z score, which was to be used alongside a weight for height Z score or a length for age Z score. And this was a metric which could look at microbiome maturity. And so by taking a child who was malnourished, they could see if its microbiome could predict whether it will grow very well in the next couple of years of life. So this was the trajectory of healthy child growth in the first 2 years of life, whereas a child who maybe wasn't growing so well had a maturity index of the microbiome maybe somewhere down here off the spline of the curve. And using this model, they were able to find that current RUTF feeding may not be sufficient to persistently restore the maturity or the health of the gut microbiome following a bout of severe acute malnutrition. So if a child came in who was severely malnourished, who had a weight for height Z score less than -3, their microbiome for age Z score was very, very low. It was down at -6. But with feeding, with standard RUTF therapy, this microbiome index tended to increase up to a normal, mature state. But after 3 or 4 months of follow-up, it tended to decline again. And so this might suggest that reductions or a return of the microbiome to an immature state may precede SAM relapse or may precede faltering growth in acute malnutrition. And so really this was a really important paper to show that the microbiome may be used to predict how well a child will grow in this early life period. And so that was a case of severe acute malnutrition, and more recently the microbiome of stunting has been characterized in sub-Saharan Africa as well. And in this study, the authors not only sequenced the stool microbiome, which was used to represent, I suppose, the lower intestinal tract, they also managed to sequence some duodenal and gastric aspirates as well. And in healthy growing kids, they found that there was a normal composition of microbes, which would usually reside in these different regions of the intestinal tract. But they found that in stunted kids, there was what they called a decompartmentalization of the intestinal tract, whereby species that usually live in the oral cavity were being found much lower down in the lower intestinal tract. And they hypothesized that when some of these commensal organisms or some of these pathobionts begin to live outside of their usual ecological niche, this may contribute to some of the chronic inflammation which may be contributing to stunting and faltered linear growth. And so we recently summarized this in a review which looked at the microbiome in the first 1,000 days and how the pathways by which it might contribute to not only this phenomenon of EED, but also of faltered growth. Whereby in a healthy child, the microbiome is really important for colonization resistance against pathogens. As I've shown before, it's extremely important for IGF-1 and growth hormone production and metabolism, and the production of, for example, short-chain fatty acids, which contribute to this pathway. But if you have a disturbed microbial community, it becomes not as good at resisting pathogen colonization. It's not as good at digesting nutrients in the diet, and it's not as good at producing some of these important metabolites as well. And thereby it leads to not only local but chronic inflammation throughout the body, which may proceed or contribute to faltered growth through disturbed metabolism as well, host metabolism. So I want to end and give you some of the evidence so far for what we have for studies that have directly targeted the gut microbiome in child undernutrition, and where we might look to the future and where such therapies could complement nutrition or WASH or some of these other interventions which we would hope could improve child growth in the long term. But we first have to consider when to intervene and when a child may be most susceptible or when their body may be able to adapt and respond to an intervention targeting the gut microbiome. And so what we've kind of tried to portray here is that the gut microbiome in a healthy child will go through this normal acquisition and cycle, which is intergenerational, I suppose. It's passed on to the infant from the mother at birth. But if there's some sort of environmental disturbance, for example, poor hygiene, poor diet, or these other factors that contribute to undernutrition, the microbiome tends to move off course. And it tends to stay off course here, and if there's a case of acute malnutrition, it tends to move a bit further off course. So we want to try and guess where is the best place to intervene in this trajectory that's been disturbed. And we think that during this first 1,000-day period in green here are very good times to intervene, because as I showed before, the microbiome is assembling, the architecture is still being built. And after the first 1,000 days, it's relatively stable. So maybe these aren't the best times to intervene after 2 or 3 years of age. But possibly prenatally in the mothers, or even preconceptionally, could be another time to intervene, because we think that the malnourished microbiome may be intergenerational. And this has been shown in animal models of more metabolic disorders associated with obesity, whereby if you feed animals a poor diet lacking in some of the fibers that the microbiome needs, it leads to this loss in microbiome diversity. And this is accentuated across generations. If the animals then are fed a healthy diet again, and then the next generation are fed this insufficient diet, the microbiome gets worse and worse and worse over generations. So we'd hypothesize that because there's this vertical transmission of microbes between mother and child, that there could be this intergenerationality of a malnourished microbiome. So there's been quite a number of antibiotic studies which arguably target the microbiome, but these have been a bit, not controversial, but they've been a bit conflicting in their results. If we look at antibiotics in severe acute malnutrition, which in complicated cases are routine, the evidence is a bit conflicting. There was a few large studies over the last few years, 2 of which found no nutritional or mortality benefits in kids recovering from severe acute malnutrition, and 1 which did find a small benefit. In terms of preventing stunting, I showed you at the start there tends to be a very, very small but significant increase in linear growth with antibiotics, which may be due to reducing the pathogen burden, or it could be due to shifts in the microbiome which alter the ability of it to digest and absorb nutrients. We know in Western settings that there's a positive association between antibiotic use in very early life and with weight gain later on. Children who have more doses of antibiotics very early on have a higher risk of obesity later on. And so there tends to be an association between antibiotics and growth. The issue of probiotics is definitely a controversial one. And this is mainly due to some differences of opinions in what are probiotics. And many probiotics that are being used which haven't shown a significant clinical benefit. A probiotic can really only be termed a probiotic if it has shown in clinical trials that it has a specific health benefit. In fact, in Europe, EFSA have said you can't even use the term probiotic anymore on any food product because it needs to have a defined clinical benefit. So a probiotic is really a very specific species of usually bacteria, but it could be another microbe as well. There was, however, a very large study of probiotics in SAM, which was published in The Lancet quite a few years ago now, which found that there was no benefit at all of this combined probiotic and prebiotic, this insoluble fiber, in improving nutritional status in infants recovering from SAM. There was a strong but non-significant trend in reducing outpatient mortality, but again, not significant. However, there was a recent study last year right here in India of a similar intervention, a Lactobacillus plantarum probiotic combined with a prebiotic, which is designed to, I suppose, feed these beneficial microbes, was given to infants in the first 2 to 4 days of life in a huge study of about 4,000 infants with the aim to reduce cases of sepsis and death. And this was successful. This probiotic intervention, which was given for 7 days, beginning on the day between day 2 and 4 of life, significantly reduced sepsis and deaths in these infants. And the authors concluded that this should be a standard treatment or a standard prevention for sepsis in low-income settings. And this was in healthy infants over 2,000 grams at birth. So it wasn't even in preterm infants who are more susceptible to sepsis. And this has now actually been replicated by giving this intervention to mothers as well in Western Africa and in Australasia as well. But importantly, there was a smaller result which showed that this probiotic intervention tended to increase weight in these kids as well, suggesting that if you do intervene very early in life in the microbiome, there is potential not only to improve the immune responses or reduce sepsis, but also improve growth in the kids as well. So I'd say watch that space because there's definitely going to be further evidence coming out from that. And I'm going to present one of Mark's slides, actually, which I don't think he pointed to yesterday, but another intervention which you could arguably say is targeting the microbiome is that of fermentable carbohydrates and some of these legumes that he spoke about yesterday, which not only contain very good protein, but also contain a lot of these fermentable carbohydrates, which are very important for feeding the gut microbiome. And they've found that these fermentable carbohydrates, namely cowpea and common bean, tend to have a small improvement in linear growth. I suppose this is promising for growth, but it's a very small effect at the moment, and we don't quite know what it's doing to the microbiome as well. And finally, I'm sure many people have heard about fecal microbiome transplants as this extraordinarily successful treatment for recurrent C. diff, which is the concept of taking a stool sample from a healthy donor and transplanting it into someone who has recurrent C. diff, and it is 95% effective, much more so than giving antibiotics. And so FMT is now being trialed in a huge number of disease states all around the world. But there is one pilot study which has just been started in South Africa only a couple of months ago by these people who run an NGO, a stool bank in Boston. They're called Open Biome. And they're running a pilot study to see in children who aren't responding to RUTF, kids who have complicated severe acute malnutrition, aren't responding to RUTF normally. There's a trial of taking a healthy stool sample and performing an FMT in these kids. And I think this is quite interesting and quite promising. Of course, we're not going to perform FMT in cases of stunting or chronic undernutrition, but in cases of really complicated SAM in kids who aren't responding, I think there is potential for completely recolonizing and changing the microbial environment in the gut, which may improve the response and digestion of foods and hopefully of growth as well. So hopefully there'll be an update of that soon, sometime in the next few months or year or so. So it's a bit of a busy slide, but what is the future for the microbiome research in the context of child undernutrition? I think, as I've mentioned at the start, we need to shift our focus a small bit away from the pathogen model. Of course, pathogens play a really important role in child undernutrition, but there are so many other microorganisms there which are really important for how these pathogens react in the body, and how they work, and also how they interact with some of these growth pathways early on in life. As I mentioned as well, we need further data on the composition of the microbiome in other regions of the intestinal tract. EED, as I said, occurs much higher up in the upper GI tract, but we don't know enough about the upper GI microbiome in kids who are chronically undernourished. If we're looking at interventions for the microbiome, we need to, in the context of probiotics, really consider strain specificity and timing. We have to move away from just taking any sort of probiotic and throwing it into a child to see if it works, because we know that just hasn't been working at all. And we need to focus on strains which we know can colonize the impaired gut and which can really improve the status of the infant much earlier on. And of course, the microbiome doesn't work in isolation, just as we know that nutrition doesn't work in isolation, and pathogens don't contribute only to undernutrition. And really, there's this interaction between all these different facets of child development, which are very important. So I'd like to thank everyone involved in our very international team in Zimbabwe and Zambia and London and Vancouver as well. And I'm happy to answer any questions.
I just thought that was an excellent presentation, and I think it's an eye-opener that now shit is gaining importance. Yeah, and we find more people saying, oh shit, rather than oh God. Yeah, and I think I open the audience for questions. Any questions? Yeah.
Thank you, sir. One question regarding your FMT, fecal microbiota transfer. So it has been found that before giving FMT, some GCSF, granulocyte colony stimulating factor, is given. This will increase the CD34 count, and this will make the FMT successful. Otherwise, it does not last for long period. So have you done this, or it is given? And number two is that which route you are using. Either it is giving through cecum or it is giving in duodenum. But we have seen that after giving this thing, at least 3 months the IBD patients become symptom-free. But larger studies are required to say that whether it is a completely curative thing or not, these things.
My response to your first question is I'm not too sure, to be honest. I know that there are a number of factors which are only coming out which can affect, we know may affect the success of an FMT. For example, the factors relating to both the donor and the recipient. I'm not too sure how that'll work in a malnourished child. The second question was, sorry, what was that again? I've even worse memory than Andrew, I think. That was right.
You gave this thing in SIBO, That is small
sorry, yes, the
intestinal
root,
bacterial overgrowth.
The root, yeah. So it can be done both ways. There's what they dubbed poop pills. You know, it can be taken orally or given via enema. And I think that it depends on the case. In this study of severe acute malnutrition, it's been given by enema. And I think both have been shown to be equally successful in terms of whether the transplant takes. But possibly in a malnourished child, it might be more sufficient to give it via an enema, and that's what they're doing in that trial anyway.
And regarding the use of the probiotics in cases of obesity and fatty liver disease, is there any trial or anything else?
There's meta-analyses of probiotics in obesity, and there tends to be a significant but very small reduction in weight of half a kilo or something like that. So that's why I was kind of really like to emphasize that the term probiotics really needs to be looked at, because so many of these studies have given any strain of bacteria which doesn't necessarily have a real functional effect that we know about. We don't know what the mechanism is, and these meta-analyses are just combining lots of strains of usually lactobacilli, which are probably just cultured from some yogurt and given in a huge form. So there is, as often seems to be the case, a significant but very, very small effect in obesity of probiotics.
Is the probiotic safe in immunocompromised patient?
It's a good question. There have been case reports of lactobacillemia, but in general, I think there's been Cochrane reviews on the topic as well, and in general, it seems to be very, very safe. There doesn't consistently tend to be any safety issues of probiotics as of yet, anyway. Even in immunocompromised patients.
In necrotizing enterocolitis, has it got any?
Yeah, well, that's almost a standard therapy now for necrotizing enterocolitis is probiotic therapy, and again, there's been meta-analyses of that, which is shown to be, very, very, very successful for preventing NEC.
Yes.
This is Satish Billa from India. Thank you for a very interesting topic of microbiota. You have detailed that the microbiota have a role if you can identify a pattern in a malnourished child specifically, and then try and intervene if that intervention would
Sorry,
happen.
I can't quite hear. Could you?
Could you speak a bit closer to the microphone, please?
Can you hear now?
Yeah.
So in your presentation, you have highlighted that microbiota certainly has a role, and there will be possibly some patterns in a malnourished child.
Yeah.
So if you are able to identify those patterns, you could try and do some intervention that might help. But in your studies, is it just a mere presence of these in the stool sample, the microbiota, is that sufficient, or is there any thresholds they have to meet? Is there what I mean is there any levels of these microbiota in the stools that would affect it?
So that's one of the drawbacks, I suppose, of microbiome research, is looking at thresholds and levels, because everything's relative. It's all a percentage of everything that's there, rather than, if you're measuring vitamin D levels in a child, you can have a threshold. And so it's all proportional to everything else that's there. So if you see an increase in one of these microbes, which is contributing to faltered growth or to healthy growth, indeed, that's only in comparison to everything else that's there. So that's why it's very important that we don't look at these in isolation, and these patterns and looking at the whole community are important. And so I think as I presented, the Microbiota-for-age Z score really addresses that quite well. That has been applied to a Bangladesh model. It was shown to be successful, but not as successful if applied to a Malawi cohort. And that, again, shows that there's these contextual differences in the microbiome. So I think I'm addressing your question in saying that, yes, it's these combination of microbes of those, as they showed, a 24 or 25 most distinctive species which were contributing to growth, and instead of looking at one or two things in isolation.
And one thing is, I'm guessing these are not that expensive to do the testing from the stool sample, I suppose?
To sequence? It's relatively expensive. It's probably about I think Canadian dollars. So US dollars, maybe $150. It depends what type of sequence. There's a couple of different types of sequencing. You can sequence the whole metagenome, or you can sequence just the 16S gene, which will give you bacteria, and that's much cheaper these days. You could get that for less than $100 per sample. But I mean, most of it is, it's all research-based at the moment. You're not going to be able to find something clinically right now if you've got a malnourished child or a child at risk of faltered growth. By sequencing their microbiome, you're not going to be able to apply something clinically to that. And we're not at that stage yet in microbiome research, and there's still a long way to go before we'll get there.
Okay, thank you.
Coffee next. I think we could take some more.
Yeah, one more question. Yeah,
Maybe a
right
few
here.
more.
Yeah.
My question,
Just introduce yourself.
okay. I am Dr. Syed Musa Emekayum from Bangladesh. My question is, you said the antibiotic has some benefit, and the previous lecture also said that a benefit for development of height, increasing height and improving microbiota. But role of indiscriminate use of antibiotic in damaging normal microbiota, and so leading to malnutrition and leading to short stature. Our observation is that mostly we pediatricians in our country use antibiotic indiscriminately. Maybe majority of the cases are useless. So we are pushing our children in dangerous situation. Maybe our role to heal children and to improve children's height, maybe we are doing bad to the children. I need your comment. And
No,
So what he's
completely
trying
agree.
to say is, we do create a lot of collateral damage.
Yeah.
So if I'm treating a sore throat with an amoxicillin,
Yeah.
I'm knocking out all the anaerobes in my gut without realizing it.
Yeah. I mean, I neglected to talk about that and antimicrobial resistance. I mean, I presume everyone's very tuned into it. And I was merely presenting the evidence that antibiotics have been used quite a lot over the last few years, and they do show a growth benefit. But of course, there's this massive, massive risk of antimicrobial resistance, and there's this kind of risk-benefit analysis. There was a huge study, which I'm sure many people will know about, called the Mordor study, published last year, which reduced child mortality by 14%, I think, by giving this mass administration of antibiotics in 3 different countries throughout Africa, thousands of kids. And so there's big debate over that, whether, you know, in the risk benefits of this mass administration of antibiotics is hugely lending to antimicrobial resistance, and of course in disturbing the gut microbiome, which is why, as someone in the field, I'm in favor of exploring some of these other interventions which, you know, tend to improve the microbiome rather than, you know, blasting it with broad-spectrum antibiotics. Have I answered your question?
Yeah, there's one here from...
Yeah, some comments about that, since we've done a lot of work with that. In the Mordor study, what was seen was significant and persistent erythromycin resistance, but no particular differences in the microbiome population. In a study from Niger with acute SAM kids, I think there, at 4 weeks, sure, there was a lot of resistant genes in the rhizostome, but there wasn't after 6 months. So I don't think that, I personally think that the fears about antibiotic resistance are excessive and hopefully aren't going to be realized on just using antibiotics in a time and in a place. And this is just an opinion, but the persistent resistance seems to be an issue with azithromycin.
Just one more comment here, because the issue of drug was brought out. The microbiota and drugs. There is so much on it. Paracetamol toxicity, chloramphenicol toxicity. The main playmaker is the microbiota.
Yeah.
I think, and most of the idiosyncrasies idiosyncrasies could be having a basis at the level of the microbiota.
Oh, hugely. I mean, there's so many drugs that we don't know their mechanism of action, and so many of them depend on that interaction in the gut with everything that's in there. So yeah, I think it really needs to be taken into consideration.
Also the extrapolation of data, because we are all different enterotypes. The Indian enterotype is different from the Western enterotype. So a probiotic that works there need not work here, because now we talk more about a community like you showed us, rather than one germ, one disease theory.
Well, that's, I mean, the field is kind of moving towards that in the concept of responders and non-responders to, you know, a medical intervention or a nutritional intervention is quite interestingly now seems to be hugely reliant on their microbiome as opposed to, you know, other factors of the host. And so, the phenomenon of responding to a drug may be dependent on the microbiome.
Any questions? We'll take some other. Yes.
Thank you, Dr. Robertson. I am Dr. Anis Rahman from Bangladesh. My question is that we know that during normal delivery, baby gets microbiota from mother's birth canal. But when the baby delivered by cesarean section, whether we suggest the mother with breast milk, we suggest any probiotics or prebiotics.
So yeah, that's been studied quite a lot. There is a big difference in the child born by C-section. Their microbiome tends to be quite similar to the mother's skin as opposed to a child born by standard vaginal delivery. It does tend to be rescued by exclusive breastfeeding. So at the moment, I don't think there's enough evidence to suggest we need to be giving probiotics to all these kids, but there's an interesting clinical trial going on in the States at the moment of vaginal swabs of children born by C-section. At birth. So children born by C-section will take a swab of the birth canal, the vagina, and rub it everywhere all over the baby to see if that can restore a standard or a normal microbiome in the kid. But there isn't, I wouldn't give a probiotic because we don't know what probiotic is what it's going to be doing and what it's important for. And I think that results of that study of the vaginal swabs could be quite interesting.
But don't you think so? The introduction of a formula feed, even a single dose, is more detrimental than introducing a probiotic. I mean, that is a better intervention than...
Well, with formula feeding, yes. Sorry, I didn't hear about formula feeding. Yes, very much. I mean, you know, breastfeeding is the gold standard for 6 months and at least. In cases of formula feeding, I mean, there's huge research going into what microbes can be added to these formula feeds, which will, you know, stimulate what's in the maternal breast milk, and which could seed that microbiome environment. But just like milk oligosaccharides and all these factors in breast milk, it's almost impossible to completely replicate breast milk in a formula feed because the microbiome can, you know, is so complex and because it can vary so hugely between mothers.
Rory, I'd like to add my personal thanks for a simply tremendous talk. Well done. Really encapsulated all that we wanted you to do. Thank you very much indeed. We'll take a break now until, if you could be back here in 25 minutes at 10:30. Thank you very much.